All right, so we will start the technology development session. And in this concurrent session, we have three brilliant scientists. I mean, they're my favorite scientists. And our first speaker is Alex Shalek. Alex Shalek is a core member of the Institute for Medical Engineering and Sciences, an associate professor of chemistry at MIT, and an institute member of Broad Institute. His lab's research is directed towards the development and application of new approaches such as single-cell genomics to study human health and disease. Dr. Shalek is very smart, and he has received numerous honors. And most importantly, he's one of the very few scientists who dare to challenge me in karaoke singing. So let's pass the microphone to Shalek. Alex.
Thanks, Guoji, and thanks for having me, and thanks for the kind introduction. I wouldn't say it was much of a challenge. I'd say you won that one far and away. So let me share my screen really quickly, and thank you all for having me here from Boston. There we go. Guoji, can you see my screen right now? Are we all good?
Yes.
Perfect.
Very nice.
Let me just get a laser pointer. All right. Thank you all for having me. It's wonderful to be with you. I'm sorry I couldn't be there in person. I always enjoy these gatherings, and I always look forward to meeting new colleagues. What I was hoping to do today was to tell you a little bit about some of my lab's recent work that's focused on identifying and rationally modulating drivers of enhanced and diminished immunity. And a lot of that is built upon the development of tools, and you'll see that as I go through my talk and talk about how problems in biology drive us to innovate in technology and vice versa. So at a high level, my lab is interested in how balance is achieved in immune responses in tissues. I'm sure I don't have to convince everybody on this call of the importance of this, particularly given the ongoing pandemic. On the one hand, over here, if you have hyperimmunity, you can get tissue damage and death. And on this side, if you have hypoimmunity with too little response, you can become susceptible to the outgrowth of opportunistic infections or the development of tumors. What my lab is really interested in is what is it that defines homeostasis in our tissues? What are the deviations that are induced by disease? And how can we think about therapeutically rebalancing systems or prophylactically buffering them in the first place? And to do this, we develop and apply tools that let us understand what these complex cellular ecosystems look like in health and disease. What this really means is, like many people in this audience, we spend a lot of our time thinking about the cells that comprise our tissues, thinking about the diversity of different cell types, the qualities that they possess, the breadth, how durable they are, and what the locations they exist in our body. Now, as I'm sure you've heard many different times, we've got about 10 to the 14th cells in our body. So the question really becomes, given our throughput, how do we think about prioritizing which ones to study? And the way that we've really been doing it is we've been focusing on phenotypes that are either naturally associated with control of disease or associated with a protective vaccination. So starting off with this idea of natural correlates. One of the systems that we've been studying extensively for a number of years now is M. tuberculosis infection. I'm sure most of you are well aware of the magnitude of this problem. It's estimated that about a quarter of the world's population is latently infected. There are about 10 million new cases a year and 1.6 million deaths related to TB, which makes it one of the leading causes of infectious disease death. I'm not sure if COVID's overtaken it yet, but very often we don't talk about it quite as much, at least in Boston, where I am, because the burden of disease really disproportionately impacts sub-Saharan Africa and Southeast Asia. Now, the interesting thing about TB is that when you look at the life cycle, you see that something strange happens. Somebody coughs, you get the spread of TB, which gets ingested, goes in and gets breathed in, goes to the lung, gets inside of a macrophage, and then it goes down and forms a structure called a granuloma, which can either resolve eradicating the bacteria, reach a détente, or where you can have the bacteria continue to grow. So you might fundamentally ask yourself, if all these different divergent outcomes are possible, what is it that leads to a granuloma that will resolve or one that progresses? Now, this is a complex multicellular ecosystem, and so what we want is an unbiased way of working with these small, difficult to use tissues. And so the technology that we've been using is one that we co-developed with Chris Love's lab called seqWell. It's a simple, portable microwell array platform for single-cell RNA sequencing. And we're basically taking these granulomas, dissociating them, capturing these cells with uniquely barcoded beads, just like in the droplet-based approaches, but using gravity, sealing, lysing, and pulling them out. And this is one of many different microarrays. Guoji's lab has obviously developed some very nice platforms, as has Peter Sims. But for this particular application, we like this because it's portable and it goes into the BSL-3, and we can use some nasty lysis buffers here in order to liberate our RNAs and neutralize our bacteria. On top of this, we're throwing in some new molecular biology that we just recently published this week in Immunity, where we can get a significant boost in the number of genes and transcripts that we capture per cell and get up almost to the level of some of the best-in-class plate-based protocols, and really get good detection of transcription factors, cytokines, and cytokine receptors, which is essential here. So we have this simple portable technology, and what this lets us do is go and profile samples that normally wouldn't be able to look at. So here we are popping down to Durban, South Africa to work with our partners, Alanian. We can take lung resections from people who have severe tuberculosis, and within exactly the same lesion, or within exactly the same lung, we can see lesions that are healing and ones that are necrotic. Now the question is, can we take this technology and get very high quality single-cell RNA sequencing data? The answer is yes. We get these beautiful t-SNE plots like you've seen before with these very nice marker genes that define each of our populations. The problem is that we don't actually understand what we're looking at. And the reason for that is that we only get tissue when somebody's very sick. And so we get the tissue, we don't know it's a granuloma, we have to go palpate it with our fingers, and then we have to profile it, and we have to hope that we're getting something new. And really, this is severe end-stage disease. So to get around this, what we've done is we've partnered with Joanne Flynn and Hannah Gideon to use this non-human primate model of cynomacaque that they've developed, where you can do low-dose infection by bronchoscopy, and you get a lot of what you'd see in humans. And critically, at these earliest time points post-infection, you follow the trajectory you'd see in humans, where some granulomas go on to have very high bacterial burden and others go on to resolve. What this means is that we can now do an incredibly well-controlled study of this, difficult to study pathogen, where we infect at week 0. We use PET-CT imaging to track the animals at weeks 4, 8, and then at 10 when we do necropsy. Take individual granulomas that we've looked at the dynamics, and then sequence them and look at what's happening when it resolves versus doesn't resolve. So the first place that we did this was looking at 4 different animals. And here we're looking at the overall amount of activity by PET. And we can see that we have 3 animals that have interesting dynamics and 1 bad monkey. But importantly, within a single animal, we can see that we have granulomas that have high bacterial burden and some that completely resolve, that sterilize the infection. Overall, it looks like we have tertiles, so some lesions that have low amounts of bacteria, some that are moderate and some that are high. But even when we look at these, we see equivalent amounts of DNA for bacteria, suggesting that the difference is in the killing as opposed to in the amount of bacteria that was originally there. And we can see that by looking at the ratio of colony forming units versus chromosomal equivalents. So we apply sequencing magic, like you've seen before, and from these 26 granulomas, we get 100,000 plus high quality cells that represent about 13 distinct lineages. We can now ask ourselves, what are the cell types that associate with bacterial burden? When we look at this, we find surprisingly that there are actually a number of cell types that associate with bad burden. So plasma cells and mast cells and endothelial cells are all highest in lesions that have high bacteria. This is strange. We never would have thought about looking at plasma cells or mast cells. And this is one of the things that's so cool about single-cell RNA sequencing. It's not just that we can do deeper phenotyping on specific cell types. It's that we don't have to make assumptions ahead of time. And so now we can find cells that we wouldn't have normally looked at. And we can go and validate by looking at histology sections. And we can see that actually around the periphery of the granuloma that I'm showing you right here, we can see positive staining for markers of mast cell populations. So what about things that associate with good outcomes? Well, there's really only one population in our data, and that's this hybrid T and NK cell cluster. But you might say, that's great. I know that T cells associate. Why did you do sequencing to discover that when you could have looked at CD3? The answer is that hidden within this cluster are actually 13 different subpopulations that range in their functions from being more cytotoxic to being more stem-like to being more Treg-like to even having hybrid states that we wouldn't previously have thought about, like type 1, type 17. So we can now go ask ourselves which of these populations associate with burden. And what we find is that it's not all of them, it's a subset. So we see that there are no clusters that associate inversely with burden, that they're all, neutral or good, but that there are a subset that actually associate with good outcomes. The strongest of which is over here on the right, which is this type 1, type 17 cluster. Now, in the past, people have seen features of type 1 and type 17 responses associated with good outcomes in TB. So you can see that, for example, that there are correlates of control that associate with interferon gamma or IL-17A, but we really hadn't decided whether or not these were within the same cell and what were the features that were associated with them. As we dive into our data a little bit more, we can see that this cluster is enriched for ROR alpha and IL-23 receptor. But interestingly, although we see RORC, which is a canonical marker of Th17, we don't see IL-17A and F. So this looks a little different than we would have expected from the literature. Moreover, when we look at this cluster, we actually see that some of the genes that we think of as type 1, like interferon gamma and TNF, only show up in a fraction, suggesting that maybe there's greater granularity here. If we subcluster, we find out that actually there are 4 different substates even within this hybrid phenotype. All of them are type 1, type 17. We see broad expression of things like DHLEH40, RORC, and ROR alpha. But we see that subcluster 1 is more CD4 in its phenotype. We see that subcluster 2 is more cytotoxic and CD8-like, and subcluster 3 is really where we get the interferon gamma and TNF. So this is the canonical hybrid type 1, type 17 by the best numbers. And now we can say to ourselves, well, are all of these good or all of these bad? And it turns out that the cluster overall correlates strongly with burden. When we look, it's actually the CD4 population, the CD8 population that are associated with good outcomes, whereas this phenotype right here that has gamma and TNF actually does not associate with restriction. Now, what could this be? Well, if we go and scan the literature, it turns out that there was this paper that came from Dick Flavel's lab at Yale that says that this phenotype right here among CD4s is associated with, is a precursor to tissue-resident memory in the lung and protects against certain types of bacterial infections, against Klebsiella pneumonia. So it's nice that we see this not only in CD4 and that there's precedent for it, but it's interesting that we also see it in CD8. Now, you might say, why do we get this cluster over here that looks gamma-like, effector-like, that doesn't correlate with burden? Well, in looking more broadly at the correlates, what we discovered was that there were certain populations that, look more cytotoxic-like. Some of them look more adaptive, some of them look more innate. And when we went and dove into the genes, we could see that really there was a separation between things that we would have thought were more innate immune responses and more cytotoxic in nature. This led us to wonder if there was some greater granularity in the dimension of time than we previously missed. And when we went back and looked at our granulomas by PET, what we saw is that actually there was some incredibly rich dynamics. There were some granulomas where we could detect them immediately by PET scan at 4 weeks, and some that really only emerged at 8 weeks. When we looked at what bacterial burden was in these, we saw that actually the original ones, the ones we detected early, had the highest number of colony-forming units, so the most viable bacteria. But if we looked at the amount of chromosomal equivalents, how many bacteria were there, we saw that it was roughly equivalent between the two, suggesting that these late lesions are actually doing a better job of controlling. We can ask what associates with late versus original lesions. And we see that these type 1, type 17 populations that came up before are actually strongly associated with this late response. So they're more adaptive in nature. And there's some cytotoxic ones that come on early. We could ask, are there populations within this cluster right here that give actually greater control? And so we can separate these things into high and low CFU. And when we look at this, we actually see that there are a number of populations that associate with the lowest CFU and the highest CFU. Most importantly, some of these innate and adaptive like cytotoxic populations appear to be good for initial control. But this type 1, type 17 response that we talked about before is no longer significant. Though, interestingly, if you dive in and you look at the subsets, it turns out that that type 1, type 17 response that has interferon gamma and TNF is. Now, I've been talking a lot about T cells and we're talking about technologies. One of the things that you might want to know is, what are these T cells targeting? Well, one thing that we recently did with Chris's lab is we developed a protocol to pull out the TCR sequences so we could figure out the clonality of these cells. It's a really simple protocol that's adaptable to many different single cell technologies that already exist. Basically, do something like SureSelect, where we use biotinylated baits to pull out the TCR sequences and look at them so that we can see how a particular clonotype relates to a phenotype. When we do this, we can see that we get TCR alpha and beta detection and co-detection across many of the cells, and enrichment in a subset of specific clusters. More specifically, really the places that we're seeing enrichment is in the proliferating T cells and these Th1, Th17 cells. We can ask ourselves now things about clonality and about the sequence of these T cells. Like, for example, where do we see sharing among the alpha sequences, the beta sequences, and both? What you can see is that actually most of these TCR sequences are private to each animal. So here I have 8 granulomas within one animal, and you can see some sharing of some of the sequences. Here I have another 8, you can see some sharing. Here's another animal, you can see some sharing, but I don't really see sharing across animals. If I look within this one right here, we have the greatest number of T cells coming out. What we see is a good amount of alpha sharing across the different granulomas, a good amount of beta sharing, and a good amount of alpha beta sharing, even between high and low burden lesions, which is interesting because it suggests that the same T cells are in both high and low burden lesions and could be targeting bacteria for killing. We can go look to see if there's a change in phenotype. And what we see is that actually there's a strong association between phenotype and clonotype. So within a specific TCR sequence, we very often see exactly the same population. Sometimes we see a shift, for example, between T1, T17 and proliferating. And that's not that surprising because, these T1, T17 cells look like they're cycling, but the assignment depends upon what the relative balance in gene expression is. But most importantly, if we go look at our high and low burden lesions, what we see is exactly the same clones with exactly the same populations, both of them, suggesting that really control within a lesion is not about the clonotype of the T cell or its phenotype, it's really about the abundance and getting more there. So if we try and integrate all of this together, what comes out of this profiling of unnatural correlates of control is that really granulomas that seem to resolve are associated with these type 1, type 17 responses and some cytotoxic CD8 responses. Type 2 responses are bad. So mast cells, plasma cells associate with these lesions that have bad outcomes. And maybe the idea is that you have a wound healing response, a type 2 response that walls off the granuloma, protecting it and making it so the T cells can't get in. And surprisingly, even though this is TB, this is very reminiscent of some stuff that we saw in the context of chronic allergic inflammation, where also mast cell and plasma cells were associated with bad outcomes. And so it's interesting to see how TB might look like allergy, but then again, one of the biggest problems with TB is that it's not really an immunogenic bug. So not surprising that we see overlap. So that's what we see on the natural control. What do we see on vaccines? Well, as I'm sure many of you are aware, there's a vaccine against TB, BCG, an attenuated form of Mycobacterium bovis. It's administered intradermally in places with high prevalence of TB. The problem is that it's only effective in some places and only effective in some cases against progression to active disease. We don't know why this is. We were fortunate enough to pair up with Bob, Mario, and Tricia, who were running a study at the VRC at the NIH, where they were trying different routes of immunization with BCG, aerosol, aerosol ID, intravenous, and intradermal. Seemed strange to me at the time, but they had a route into a secret message, because when we saw the outcomes of the data, it was incredible. So unvaccinated animals had very limited control when they got challenged. But when you took rhesus macaques, which aren't normally protected against TB, and you vaccinated with intravenous BCG, all of a sudden you got sterilizing immunity in 6 of the 10 animals, 3 that only had 1 granuloma and 1 that was a bad monkey. And so you might say, this is fascinating. What is it that associates with IV BCG? What is it that this vaccine leads to that actually provides protection relative to the more conventional routes? Well, we ran a lot of the same magic. We did some bowels, bronchial alveolar lavages, looked at what were the responses that showed up. And once again, we saw convergence towards type 1, type 17 responses, really now in CD4 as opposed to CD8, though that may be a factor of some of our studies, and these M1-like responses. And interestingly, ID vaccination induced a strong response. It just was not the response that we wanted. And I'm happy to talk about that more, but given time, I want to wrap into this bigger question of what can we do with these signatures? How can we leverage them in order to develop better therapeutics or prophylactics? Well, here, my lab is really taking a nod from a number of beautiful studies that have been done by people in immunotherapy, where basically what they're doing is rationally pushing and pulling on different cellular pathways to modulate immune cell function. And so the first place that we tried to use our single cell data towards this end was actually not in TB, it was in HIV, where when looking at elite controllers, individuals who can naturally suppress HIV without antiretroviral therapy, we discovered a specific phenotype that I'm showing you here in cyan that kept on coming up in our single cell data. We found surface markers for it, CD64 and PD-L1, and it turned out it was specifically induced in dendritic cells from elite controllers. It correlated with CD4 abundance and negatively correlated with viral load. So it looked like it was associated with control of virus. Now, once again, this is a place where elite controllers can make it, which is great, but we wanted in chronic progressors for therapeutic or in healthy donors for prophylactic. So what we wondered is if we could mine all the existing perturbation data to figure out which perturbations would most strongly induce this phenotype. And that might suggest that we were pushing more cells in that direction. So if we look at our data, from looking at some TLR stimulations, we can see that the thing that is the most right shifted over here is a TLR3 stimulus, poly-IC. And what this suggests to us is that actually, if we took a healthy donor and we added virus in vitro plus TLR3, we should get a response that looks like an elite controller. Turns out this works beautifully. You can take PBMCs from healthy donors, add virus, you'll get a small boost in this fraction of cells. You add TLR2, 4, 6, or 8, you'll get a little bit more, but it's really with TLR3 that you get a significant boost in the number of these cells. And we went on to show that these dendritic cells drove the same polyfunctional T cell responses and greater proliferation. Now, here we were using TLRs. The question is, how far could you take this? And we wanted to know if we could repurpose existing perturbation data sets. So we took some nanostring data that had been done on shRNAs in mice, and we ranked these different perturbations by how much they impacted the population we wanted relative to the others. This gave us a volcano plot that looked like this, where now we have a number of different transcription factors and signaling molecules we think are important for the phenotype we want. We see hypotheses that come out of here, like that TBK1, a kinase, should be something that if we inhibit, will downregulate this population. We can test that in elite controllers, and it turns out this is true. If we add this kinase inhibitor, we can downregulate the fraction of cells making this response. And you might say, well, why would you want to do that? Well, in some cases, aberrant dendritic cell responses can actually lead to hyperimmunity. So is this framework applicable to TB? Well, we can go look at monocyte states or macrophage states in our granulomas that were associated with good or bad outcome. We can see that there's some that are restrictive. We can go ask what are the signaling pathways that show up there. Here it's NF-kappa B, and then we can go directly target them and show that actually we can significantly increase the amount of bacterial burden. So we now have a way of pushing and pulling on these systems. So to conclude, what I hope I really showed you here is that TB is this incredibly heterogeneous disease, both between and within hosts, that we can use different models to recreate essential features, and that if they're selected appropriately, they can help us to accelerate interventions back into humans. And a lot of what my lab has been doing over the last little bit is trying to study human biology, to take it into model systems, and then to think about how these models will enable us to recapitulate features so that we can bring things back to humans. To do this, we've been developing a lot of large computational libraries of data and ways of translating between them. So we have a resource called Alexandria that we've stood up that basically brings together lots of different data sets so that we can query new things coming in. And we keep on trying to power this with more data from models and from different perturbations systems so that we can discover what's most important. I'll just close up really quickly by saying that we keep on trying to develop technology that's inspired by a lot of what we do here. The one thing I'm going to share very quickly is this thing that we've now developed to make some of our clinical processing of our SQL arrays easier. Where now, instead of trying to run all the way through the protocol, we load the arrays, snap freeze them, we can even ship them and then come back and look at them later. We did this in the context of liver FNAs run in Rotterdam fresh or frozen in Rotterdam and then shipped to the US for processing. We saw that we got equivalent cell capture in both technologies, whether we were using fine needle aspirates from liver or PBMCs. And that most importantly, when we looked at our individual populations, that there was no differential expression between the samples that were run in Boston or in Rotterdam, suggesting that you could run these complex samples, archive them, and then process them centrally up to a few months later or whenever was convenient and get equivalent data. With that, I'm going to thank my lab. They're awesome. And in particular, I'm just going to thank Travis right here and Mark, who did a lot of the granuloma studies, who's back here. I'll thank a lot of my collaborators who don't have time, and I'll thank all my funders who have been fabulous and hopefully will continue to support us. And thank you, and apologize for running a minute over. I see that I'm at 21. And I'll pop out. Thanks for having me, Guoji.
That's a great talk. So any questions? I mean, for the audience, you can ask questions in the Q&A window. And for the plenary, you can ask questions just by turning on your camera and microphone. All right, maybe I should start with a question. So regarding the like well-based technology versus a droplet-based technology, can you comment on the differences? And is there any difficulty integrating data from both technologies?
So it's a great question, Guoji, and I'm sure you've seen a lot of this in your own technology as well. We don't have a hard time integrating droplet or well-based approaches when they use similar molecular biology. So we find that DropSeq and seqWell, for example, integrate very well because it's the same beads, the same molecular biology in a lot of ways, as it comes forward relatively seamlessly. There are populations that you do a better job of capturing on wells than you do with droplets because the encapsulation actually will burst some cells. So for example, we see that granulocytes, hepatocytes, things of that nature come in a little better in our data. But for most parts, we see that the data is equivalent. Now, if you try and integrate seqWell or DropSeq data with 10x, there are some things that change a little bit. And so you have to think about that. And there are differences in the amount of ambient RNA contamination. But really, what changes what we get in our data is the molecular biology. So the way in which we do the reverse transcription, the addition of a second PCR handle, the amplification, more than just about anything else. What do you see in your data, Guoji?
Yeah, I mean, we're still trying to figure out, particularly, I mean, we want to integrate high throughput data with, high precision data and there are always like difficulties. And also, one issue I realized is like when we try to map single cell data with traditional fax data, there's always discrepancies, particularly for T cells. And in T cells, like the traditional classification CD4, CD8, I don't think that clustering is like the most dramatic variation within T cells.
It's definitely not. And actually, one of the interesting things in the data that we see is that very often we get very clean clusters that are both CD4 and CD8. And one of the most interesting things about the TB is that actually the functional phenotype over conventional lineaging is what dominates. And then there are interesting things like sometimes you have cytotoxic CD4s where actually they behave just like CD8s. So I think that in extreme circumstances, you can very often see that both populations will behave similarly, even though they might have different peptides that they're recognizing. I see one about SNVs from RNA-seq data. It's a great question. You can look at single nucleotide variants. Different protocols have different efficiencies in this. And the more copies of the transcript, the better you are at doing this. So a lot of the protocols, like the high throughput protocols, do 3' end reads or 5' end reads. And so it is usually pretty good for looking at single nucleotide variants that are close to the 3' end or the 5' end. Like with the TCRs, you can build targeted sequencing libraries to pull down specific transcripts and look for specific mutations. We published a paper with Brad Dornstein's group in Cell, that's Van Galen et al. In 2019, where we did this to actually, link clonotype to phenotype, or their, cancer clonotypes. But, in general, if you want to look at many single nucleotide variants, then you usually want to do it by using a full-length method like SmartSeq, where you do it in plates, because then you'll get better coverage of the entire transcript. I can go from there. But for simple applications, you can even do it off these 3' methods or by building targeted sequencing libraries.
And we have one question. What do you think of calling SNV from scRNC data? SNV, single nucleotide variation.
Yeah, as I just mentioned, I really think it comes down to getting enough coverage over the region of the RNA that you're looking at. And so, you have to figure out where it is relative to, either the 3' or 5' end if you're using massively parallel sequencing. You can sometimes build targeted sequencing libraries to look next to a site where there might be a variant, or you need to use a full-length protocol so you get it. But obviously your confidence in calling those variants goes up for more highly expressed genes, where you get more copies with more barcodes that you can make sure that it's not a PCR error, much like when you were trying to call other approaches. So I see one more there, comparing single-cell RNA-seq data and facts. Are you able to use the transcriptome to improve the discrimination of aberrant/cell populations obtainable by facts? The answer is yes. I don't have time to show you here, but we've done a lot of things where in cancer, where we see aberrant phenotypes, for example, in acute lymphoblastic leukemia, where conventional surface markers don't actually discriminate well. We can not only figure out what are the gene expression programs that define cells at different aberrant developmental stages, but we can use that for doing better classification of those cell types. Now, I want to be clear that the phenotypes that you get out of single-cell RNA sequencing are different than the phenotypes that you get out of histochemistry or FACS, because there isn't always good agreement between RNA and protein. But you can get very good phenotypes for some of these cells from RNA sequencing. Identify rare cells. I see that one more thing right there from Kathy. And the answer is yes, but it helps to know what you're looking for, because in a lot of places, the way in which you analyze single-cell RNA sequencing data is you look at variation over all 20,000 plus dimensions. And then, if there's only one cell that comes in your, say, 10,000 cells, it's going to contribute a very small amount to your overall variation, so it might not come through. If there are particular phenotypes you care about, like stemness, you could look at cells over stemness and you might be able to discover that there's one cell that looks very unique with respect to that. So really, if you want to think about rare cells or rare phenotypes and sort of this unbiased data, you have to approach the data analysis a little different. And there's been some really nice work by a number of people that have focused on how to pull rare cells and rare markers for cells out of your sequencing data, but it's a slightly different tack to the way in which you think about looking at existing single-cell RNA sequencing data sets. And so like race ID would be a good approach there.
Okay, we have another question. Is SQL platform commercially available for users? I think he's asking whether in China it's commercially available. Is it?
So we started a company to sell it. The way that I think about it is very differently. I would like everybody to be able to use it. We have put everything up online for researchers. You can copy all our protocols. There are videos. You can get on usually a plane, but now just Skype, and we will teach you to make these things. We will teach you to load them and run them. Really, what we're trying to do here is to empower the global scientific community to run these sorts of analyses and really to work with complex human samples that you normally couldn't work with to make inroads into disease. So you can run it right now. You can build it yourself. If you don't want to build it yourself, the Koch Institute at MIT will sell you a chip for, I think, about $50 a piece, so not very expensive. And we have a company called Honeycomb that is about to start beta testing where these are going to come together as snap together plastics. And they look really sweet. I hope they work as well as I think they will. And then it's just going to be this really easy snap together system where you should be able to do everything that we're doing now, but with all the difficulties.
Okay, thanks, Alex.
Thanks, Goji. Thanks for having me.
Thanks.